Hi, my name is Jenny Persson. I'm a professor of tumor biology from Umeå University in Sweden. The title of my talk is Fc-gamma receptor 3A interplays with AR and PIP5K1α to promote growth and metastasis of prostate cancer. The aim of our study is to study the role of immune cell receptors in cancer growth and metastasis. Our first focus is to study the role of Fc-gamma receptor 3A in prostate cancer progression, and we wanted to further identify whether Fc-gamma receptor 3A may be used as a therapeutic target to be used as an anti-tumor microenvironment therapy. As you can see here, Fc-gamma receptor belongs to the Fc receptor family. These classes of immune cell receptor facilitate its function in immune surveillance and immune cell defense systems. The Fc receptor mediates cytotoxic killing and can eliminate the pathogen as the Fc receptor is expressed by NK cells. And this receptor can be used by NK cells to recognize the pathogens. And the activation of Fc receptor can allow NK cells to facilitate cytotoxic activity to eliminate pathogens. So one of the hallmark of cancer cells become invasive and metastasis is that cancer cells are infiltrated or surrounded by the cancer associated stromal cells, and these cells consisting of infiltrating immune cells, fibroblasts, and other stromal cells. So the defects in immune surveillance is associated with cancer metastasis, allow cancer cells to gain the invasive ability. So as you can see in this picture, the cancer cells are in gray color, are surrounded by the immune cell, various types of cancer-associated immune cells that are local bone marrow-derived stromal stem and progenitor cells, and also cancer-associated fibroblasts, endothelial cells, parasites, and inflammatory cells. And these cells will allow cancer cells to gain the ability to invade and also form angiogenesis. So in the case of immune cell and also the immune cell receptor related to the Fc receptor, what we have already observed is that in myeloid cells and T cells and NK cells, they all express the Fc receptor, and this Fc receptor may be used for enhance the immune tumor antigens, as illustrated in this picture. So our hypothesis is that Fc-gamma 3 receptor may be expressed by by the cancer cells, as what you can see here. The cancer cells utilize the FcγRIIIa receptor to interact with the cancer-associated myeloid cells, which also express high level of FcγRIIIa receptor A. By interact or physically associated with each other, cancer cell may utilize the myeloid cells or inflammatory cells to facilitate the signaling transduction to enhance this cancer-associated factor lipid kinase PIP5K1α to induce the activation of the phosphorylated AKT, which is a master regulator to promote proliferation, survival, and invasion of cancer cells. So therefore, we wanted to first understand or examine, is FcγRIIIa receptor 3A expressed in primary tumor cells and metastatic lesions from patients with prostate cancer? Are the elevated levels of FcγRIIIa receptor 3A protein and mRNA in prostate cancer associated with poor patient outcome? To answer these questions, we first examined FcγRIIIa receptor 3A protein expression in the BPH, the benign hyperplasia cancer, prostate cancer, metastatic lesions from bone, lymph node, and the lung. And what we found is that FcγRIIIa receptor 3A expression is significantly higher in metastatic lesions compared with primary cancer. And that is also the case for mRNA level. So we also examined and found FcγRIIIa receptor 3A expression is related or associated with poor patient outcome, as shown in this survival curve. Patients with high level of FcγRIIIa receptor 3A suffered significantly worse disease-free survival compared with those with lower level of this protein. So further, we have found that FcγRIIIa receptor 3A gene amplification is a frequent event in prostate cancer from large patient cohorts. And further, the elevated level of this gene is significantly correlated with gliosine grade. The elevated level, the higher level, is observed in patients with higher, more than 8 gliosine grade category. So this indicates that FcγRIIIa receptor 3A abnormal expression is associated with poor patient outcome. So after examined its expression with clinical outcome, we wanted to further study the underlying molecular mechanisms that mediate the action of this immune cell receptor in prostate cancer progression. So since androgen receptor and lipid kinase PIP5K1α pathway play a key role in prostate cancer growth and progression, so we looked at the expression level of Fcγ receptor 3A in association with the proliferation and expression of androgen receptor and PIP5K1α. We induced the overexpression of Fcγ receptor 3A in one of the castration-resistant cell line, that is C4-2 cells. We found that overexpression of Fc gamma receptor 3A resulted in the increased tumor spheroid formation. There are significantly higher numbers of tumor spheroid being formed by these cells, overexpressed Fc gamma receptor 3A. And also overexpression of Fc gamma receptor 3A induced increased expression of androgen receptor and PIP5K1α. So silencing Fc gamma receptor 3A conversely leads to decreased expression of androgen receptor and PIP5K1α, as what you can see here. And we also found that there's a protein-protein complex formation between Fc gamma receptor 3A and PIP5K1α, suggesting that there is a directly interaction between this receptor and the key factor PIP5K1α. And that is the important observation showing that the important role of Fc gamma receptor 3A in prostate cancer growth, and this association with a key factor that is regulating cancer cell proliferation. And this is another cell line PC3. In this cell line, we have also shown overexpression of Fc gamma receptor 3A induced tumor spheroid formation and also increased cell cancer cell ability to migrate and to invade. So this suggests that Fc gamma receptor 3A play an important role in promoting cancer cell migration and proliferation in both C42 and PC3 cells. Both are castration-resistant prostate cancer cells that are very invasive and have metastatic potentials. Similarly, we have shown that silencing Fc gamma receptor 3A also leads to the inhibition of the tumor formation, tumor growth, as shown in tumor spheroid assay in PC3 cells. And further, we have shown that there's a significant correlation between lipid kinase PIP5K1α and Fc gamma receptor 3A. So both proteins not only form complex but also it can act as the cofactor as shown in this luciferase analysis. The Fc gamma receptor in combination with androgen receptor can also induce the promoter activity of downstream target cyclin A1. That is the key cell cycle factor. And we further show that Fc gamma receptor 3A alone is able to promote the downstream target promoter activity. So suggesting that this receptor mediates the androgen receptor and the lipid kinase PIP5K1α to promote downstream target gene activation. So in order to illuminate whether the androgen receptor is directly associated with Fc gamma receptor 3A, we wanted to examine whether androgen receptor can regulate Fc gamma receptor 3A mRNA and protein expression. So in this experiment, we overexpressed androgen receptor in non-invasive link-up cells, the prostate cancer cell line that is less invasive, and we examined the effect of androgen receptor overexpression on Fc gamma receptor 3A. So here we have shown that overexpression of androgen receptor significantly induced Fc gamma receptor expression. Conversely, inhibition of androgen receptor using its inhibitor enzalutamide also led to the decreased Fc gamma receptor 3A expression already at 12 hours. And this effect prolonged to 24 hours, suggesting that androgen receptor may act upstream of Fc gamma receptor 3A. So in order to examine the functional aspect or consequences of elevated or the abnormal expression of Fc gamma receptor 3A, we first silenced the Fc gamma receptor 3A in prostate cancer cell line PC3M cells, and we induced the tumor by subcutaneous implantation into the xenograft mice. As you can see here, the PC3 tumors carrying the SI Fc gamma receptor 3A had much less volumes, significantly less tumor volumes compared with the control cells. So this suggesting that silencing Fc gamma receptor 3A delayed the tumor growth, and suggesting that Fc gamma receptor 3A play important role in tumor growth in vivo. And we also examined the tumor marker, proliferation marker Ki67, PIP5K1α, and its downstream phospho-AKT, and the invasive marker MMP9. And all these markers showed that the expression of these markers are significantly reduced in tumors expressing the SI Fc-gamma receptor 3A. We have also found that the control tumor have also shown the metastatic lesions in lymph nodes. However, the Fc-gamma receptor 3A silenced the tumors, not show any metastatic lesions in lymph nodes. Further highlight the role of this receptor in tumor progression and the tumor metastasis. So in order to examine whether Fc-gamma receptor 3A can be the targets that can be used for treatment of invasive prostate cancer, so we used the in vivo models, and as you can see in C, panel C, we use in vivo models. The xenografted tumors in mice, we allow the tumors grow into large volumes. Then we started the treatment using the monoclonal antibody to Fc-gamma receptor 3A, that is M3G8. And we use the control antibody also to treat the tumors. So as you can see, the M3G8 treatment of the mice suppressed the tumor growth, in contrast to the control antibody, which has no effect on tumor growth. And we also found this on-target effect of the treatment. The lipid kinase PIP5K1α and VEGF receptor 2 showed significantly downregulation in the tumor treated with antibodies. So in order to examine whether Fc-gamma receptor 3A can be used as also the target and the antibody against Fc-gamma receptor 3A can be used for treatment of metastatic prostate cancer in vivo models. So we first used the tumor spheroid models. We allow the tumor spheroid to grow in 2 sites, and then we implanted 40 tumor spheroids subcutaneously in mice and allow the tumor to grow large and to facilitate metastasis to the bone and to distant organs. So after the detection of cancer metastasis, we started the treatment using M3G8 antibody. So after 15 days of a treatment, daily we administrated the antibody daily, and then we surprisingly found that the tumor metastatic burden was significantly reduced in mice that were treated with the antibody M3G8. We also shown that the metastatic lesions were absent in cancer cells. This is the bone marrow metastatic lesions in control-treated group. In M3G8 treated group, there's no metastatic lesions been found in bone marrow and in other organs. So further, we have examined whether combination of PIP5K1α inhibitor together with M3G8 may have a better effect in preventing or inhibition of migratory ability of prostate cancer cells. So these results showed that a combination treatment using these two agents has a significant effect compared with a single agent. So this finally, this slide showed that our hypothesis indeed has been validated and proven, and that in this model, FcγRIIIa receptor 3A is expressed by the cancer cells, although it is also expressed by the immune cells. This receptor facilitates the interaction of a cancer and the myeloid inflammatory cells, and the cancer cells utilize the interaction and to activate the cancer cell specific signaling to promote proliferation, survival, and invasion by mediating the cancer cell specific pathways. So finally, I would like to acknowledge people in my group who did a great work for this project, and also our collaborators and the funding agencies. Thank you for your attention.